and that's why it took a little while. Good evening, ladies and gentlemen. Carestream Molecular Imaging is proud to present this webinar on multimodal optical and nuclear imaging. Presenting today will be Dr. Walter Akers, who is Assistant Professor of Radiology at the Optical Radiology Laboratory at Washington University School of Medicine. If you haven't done so already, please mute your phones. We do have a number of people in today. If you have any questions during the program, please type them into the chat box to your right, where it says Send to, choose host and presenter. At the end of the presentation, there'll be a Q&A session. We will try to answer as many questions as possible during this time, and we'll contact you directly if we're unable to answer your questions online. And with that, I am pleased to introduce you to Dr. Akers.
Hi. As Meryl mentioned, I'm Walter Akers. I'm here at Washington University School of Medicine, and mainly I've been working with optical imaging for the last few years, optical imaging of cancer and cardiovascular disease, and we've recently gone into multimodal imaging, so expanding from optical into other modalities as well. So I'm going to present some of our recent work and also some of what we've done specifically with the Carestream instrument. So what I want to discuss first is, so with the use of biomedical imaging, we can use different energy spectrum and different modalities to capture the biological events non-invasively, ideally, so that we can see more what is happening in the natural environment. And there are multiple modalities using energy from the electromagnetic spectrum to capture these data. As I said, I'm mainly focused in the visible and near-infrared optical regions, which we've been working with for a while, and now we're advancing into the X-ray, and which we've been using for a while as well, and into nuclear imaging, which uses gamma rays for capturing the data. Now, also, if you have any questions during the presentation, you can type into that chat box, and I will try and keep an eye on that to see if there are questions during the presentation. Just a moment. Okay. So initially, imaging using X-rays has been around for quite a while. Röntgen discovered X-rays and took one of the first X-ray images back in the 1800s. And X-ray contrast results from the absorption of the X-ray radiation by the biological tissues. And the X-rays do travel in a straight line from the path, from the source through anything in its path to the detector, where it's then detected by either film or by scintillator, such as sodium iodide crystals or other newer materials, which produce luminescence upon absorption of the X-rays, and that luminescence is detected by a photodetector, such as a photodiode or a CCD camera. Computed tomography is an advance of that method, which uses the X-ray source and detector rotating in the XY plane and then advanced in the Z plane, so that you can have slices which can be reconstructed into a 3D picture. In this picture, I'm showing that the skeleton has the highest contrast, and we can easily see the skeleton of this mouse, and the soft tissue contrast is less, and that is dependent on your settings of the X-ray system. But you can adjust that to see contrast as well. Well, here you can see the femurs, the bones, and the soft tissue contrast on the sides here. So these are the back legs, here's the bladder, and here's the tumor. But you can see that the tumor doesn't have really any different contrast than the muscle. And that is one of the difficulties with X-ray contrast in trying to identify lesions that may not look different than other tissues. So nuclear medicine uses the same kind of methods in a way that uses radio active reporters as contrast agents in the body. You can direct those using molecular targeted agents, or use the reporters based on metabolism or other factors to identify your disease, your lesion of interest, which is then captured either by planar scintigraphy, so a scintillator pad in just one or two dimensions, or using tomographic methods such as SPECT or PET. Like the X-rays, the gamma rays and positrons travel in a straight path, so they're relatively easy to reconstruct the images based on different methods. So the tissue does absorb some of those photons as well, but the ones that are not absorbed do travel in the straight path, and then they can reconstruct to find out exactly where they were to begin with. Now, optical imaging uses light rather than the ionizing radiation, so it's safer to use in that respect. You don't have to deal with radiation safety problems, and it's not harmful to biological tissue in general. You can also use intrinsic contrast, such as protein absorption by the hemoglobin, other compounds or chemicals that are within the tissue normally, or you can use exogenous contrast agents that enhance the contrast, specifically when you're looking for physiologic events or molecular events for molecular imaging. Now, these exogenous contrast agents can include light-absorbing compounds, also called chromophores, such as methylene blue, which is used for visual identification without any other instrumentation. Methylene blue is used for sentinel lymph node or lymph node identification, and can be used intraoperatively just through visualization with your eyes. Fluorophores, or compounds that re-emit light after illumination, can be also used, and they can be detected using optical detectors, again, such as photodiodes or digital cameras, using wavelength limiting filters. So because the compounds absorb light and then re-emit at a longer wavelength, the excitation and emission can be separated using optical filters. Now, the difficulty with optical imaging is that absorption and scattering are wavelength dependent, and those are the major factors in attenuation of the signal. Now, absorption is specifically high in the visible wavelengths by proteins, melanin, and hemoglobin, and then in the longer wavelengths by water and collagen, so that absorption can be minimized by using specific wavelengths. Now, scattering, despite this diagram, is considered relatively flat across the spectrum, but that is the main difficulty in optical imaging, which prevents depth resolution to a serious extent. Using fluorescence imaging, the diffuse nature of the light propagation, so because there's so much scattering, the light is considered diffuse, meaning that the origin of the light is difficult to determine with high resolution as the penetration depth becomes larger. So in 6 samples, the resolution scales basically exponentially with depth, so that at very low depths, such as microns, as in microscopy, you can get very, very high resolution, in fact sub-nanomolar. But as the tissue becomes thicker, such as millimeters or centimeters in biological tissue, the resolution can degrade significantly. But despite the limitations of this, fluorescence imaging is very sensitive and has a lot of advantages that are not offered by the other modalities. For example, the fluorescence signal can be long-lasting because the signal is dependent not on the decay, as in radionuclear imaging, but on the presence of the fluorophore and the excitation. So the signal can be regenerated just by exciting the fluorophore again and getting the fluorescence. So as long as the fluorophore is present, you have the signal. Also, I'm going to discuss activatable fluorescence. The fluorescence can be quenched and made silent and then regenerated by different mechanisms. Also, fluorophores can be sensitive to their environment, making biosensors capable by using sensing of some of the molecules. So the fluorescence intensity can increase or decrease relative to their environment, and using that is not the case with radionuclides. So as I mentioned earlier, the absorption of light is wavelength specific. So you may have heard of near-infrared imaging. So near-infrared light means the light in the range between, say, 700 and 900 that has the lowest absorption in most animal tissues. And that is because the hemoglobin peaks around the 500 to 600 range, and the visible wavelengths have more absorption. So the signal that is emitted is much lower when you're using visible wavelengths versus the near-infrared. But still, the imaging is dominated by scattering. And here, as an example, we have a laser excitation pulse at the surface of some tissue, and these lines are kind of demonstrating the random lock pathway of the photons, and these are just examples of scattering within the tissue until it finally reaches your target, and that target fluorescence occurs isotropically in general, and then eventually makes its way, some of the photons make its way back up to the surface and are detected. This is throwing away a decent amount of the signal that is just lost in different directions, but because you can provide a large amount of excitation and the detectors are very sensitive, you're still able to detect signals up to tens of centimeters below the surface, and that's at optimal conditions. So fluorescence imaging is generally accomplished by a couple of different techniques. The first and the fastest would be the planar reflectance imaging, and that's mainly what we're going to discuss today. And that planar means that it's two-dimensional. Reflectance describes the geometry of having your detector, in this case a camera, on the same side as your sources of illumination. So the illuminate the surface of the subject uniformly, ideally, and the fluorescence agent then is detected based on the signal and the location, and that can be very high throughput, and the cameras can acquire fluorescence images in very high rates, depending on the signal strength. Diffuse reflectance imaging, again, reflectance, the source and the detector on the same side, but it uses diffuse imaging so that the source and the detector are at separate locations, and the fluorescence is measured away from the source, and that provides better depth sensitivity because the light is not being reflected to the detector directly from its incidence, so that the background from your source is less, and so that gives a better depth sensitivity. Even better depth sensitivity is through tomography using transillumination, so the source and the detector on different sides of the subject, and that can provide 3D tomography, and is the most quantitative as far as fluorescence imaging goes. There are a lot of things for molecular imaging and physiologic imaging in cancer, including different cell surface receptors that are involved in growth signaling or angiogenesis, such as somatostatin receptor and alpha V beta 3 integrins, and then apoptosis can be detected by increases in caspase-3 enzyme, which is integral to cell death, programmed cell death. And other mechanisms can also be investigated. Molecular imaging agents generally use a targeting agent of some sort, and what we work with a lot are antibodies and then small peptide receptor ligands in general, which are smaller versions that have high affinity for example, cell surface receptors. Antibodies are very high affinity and provide high specificity and can be developed toward about any antigen, but they are generally more expensive, have a longer circulating time, and can have other adverse effects. Now, the long circulating lifetime means that the background signal is going to be high, so that the time of imaging is generally 2 to 3 days after injection. The smaller peptides, in general, have less of the adverse effects and faster imaging times, but they may not have as high affinity or specificity, and can also, because some of those do affect the cell signaling cascades, they can have biological activities if you give them at higher doses. So, for example, Integrins, as I mentioned, alpha V beta 3 integrin, it plays a role in tumor invasion as well as angiogenesis within cancer. These cell surface receptors are overexpressed in many of the cancers, and we can use a small peptide sequence based on the RGD motif to develop near-infrared fluorescent imaging agents. So on to the multimodal imaging, which we'll discuss with the alpha V beta 3 targeting agents in just a moment. The multimodal imaging is based on the synergistic combination of imaging modalities. And what's been done so far, and it was working very well in the clinics, is using SPECT CT or PET CT. So the combination of radionuclear imaging with the traditional X-ray computed tomography. And that way the nuclear imaging data can be co-registered directly with the anatomic imaging done by CT. In development are methods to do PET and MRI combined, so a single system that would do both. And then many companies are working on other methods to combine optical with CT, and then we are working with the contrast agents to combine the optical and radionuclear, so which we'll discuss. So there are a few different mechanisms to use multimodal imaging contrast agents, either by separate agents, so injecting agents that have different functions, such as looking at vascular permeability with one agent and then molecular imaging with another to compare those factors within the same animal. What we have developed are single molecule multimodal imaging agents, and that allows the simultaneous or follow-up imaging of the same probe, so that radionuclear imaging with SPECT or PET could be performed, and then because of the long-lasting signal of the fluorescence imaging could be performed after that, and for longitudinal studies, so that multiple injections of the radionuclear contrast agents is not necessary. Also being developed for preclinical imaging are genetic recorders that combine the fluorescent proteins and other genetic recorders with receptors and enzymes such as the HSV1-TK that is specific and phosphorylates the radionuclear imaging agent for PET imaging based on the genetic recorder. So for what we're going to talk about today with the multimodal imaging, we have several different modalities shown here of the same animal, and these pictures were taken with the Carestream system, showing the white light image of the mouse with the tumor here circled, because you can't really see exactly where the tumor is just based on the white light. or even with the X-ray. So we have an X-ray image, digital X-ray captured with the system of the same mouse. That it is difficult to see, but you can use the X-ray to show the anatomy, particularly the skeleton, but also you can see where the lungs are and the internal organs. With near-infrared fluorescence of this imaging agent, which was injected by the tail vein, you can see that the whole body of the mouse is fluorescent at this time point, and you can see that this agent is clearing through the kidneys and has accumulated within this tumor as well in a receptor-specific mechanism. Scintigraphy with a completely separate radionuclear imaging agent, this is NGM-111, shows the mouse signal from the radionuclear agent using the scintigraphy pad, the same pad that was used to take the digital X-ray. And you can see that there is signal from the kidneys and the tumor, but without the other anatomical information, particularly the scintigraphy, does not give any reference to what really is causing the signal. So therefore, co-registration with these other anatomic modalities is essential in determining what is causing the signal, at least non-invasively. The traditional way, of course, would be to then kill this mouse and take out the organs and measure the organs separately to determine what is causing the signal based on a percent injected dose per gram. But this can be this tumor is a subcutaneous tumor, so that it is not in the lung. It is on the back of the mouse, so it's beneath the skin. That's another good example. This is a 2D two-dimensional image. It's a planar image. So one method would be to then turn this mouse on its side and take another image, which is commonly done, of course, in clinics with the X-rays, to get the anatomic image from a different angle and then see where the tumor is. Of course, the subcutaneous tumors are easy to detect, so that I knew where this tumor was after I took the picture, and I'm confident that this signal is coming from the tumor. And in fact, this animal was sacrificed after the imaging, and those data confirmed as well. But you can see that this can all be accomplished non-invasively in a living animal, and over time to see that these agents are accumulating within the area of interest without having to sacrifice a lot of animals. And so this can speed research and decrease the cost and the animal burden, so that we're using less animals in research by using non-invasive imaging techniques. So we can also use that example was with separate agents that were targeted to the same cancer receptor. And in this recent example, a monomolecular imaging agent was developed that had a targeting group. Here's the cyclic RGD sequence with a radionuclear chelator, and using NGM-111 again, and a near-infrared reporter, all on the same molecule. And this combination allows, as I said, co-registration of the signals. And this can be used in the clinical method of, we could foresee using the radionuclear imaging initially to locate where the tumors are, so for whole body imaging, and then to follow that up with the optical imaging of those areas that are identified for example, tumor response, or for other mechanisms that would be related to our study or our clinical purpose. This can also be used for instrument development, because we know exactly where both signals are coming from. We can use that to, say, improve the optical imaging methods to be more quantitative, because we know that the radionuclear signal is a quantitative signal. So this combination can be used to monitor the fluorescence after the radionuclear imaging, and to monitor processes after that imaging. So here's another example of using a mouse that has two subcutaneous tumors, and this single agent was labeled with indium-111 and injected via the tail vein again. You can see the fluorescence signal from this mouse, and then the radionuclear signal. Now, in this case, because the optical agent has a larger molecular weight and may not have had as good a sensitivity and specificity for this tumor, the scintigraphy image is not quite as good. We can still see where we are getting signals where the kidneys line up, but also because this agent is more hydrophobic, it's cleared more via the liver, and we get a large signal from the liver contrast, even though it's on a different side of the mouse and not seen as much in the optical imaging. You can somewhat see that the liver is coming from this area, but because the optical imaging does not penetrate as deeply, you're not able to see the whole liver in this 2D optical imaging, where the entire liver is giving signal in this scintigraphy. So you can see that there are areas here that are giving signal that correspond to the tumor location. So we did make this contrast agent. This is from Sam Attilafu's group, which is he is the director of the optical radiology lab, and this agent was made here in our lab. Another method, as I mentioned earlier, is using activatable fluorescent probes. So activatable fluorescent probes, often the most typical example is using a quenched probe, which can use two fluorescent agents. So if, for this example, for simplicity, when fluorescent agents are in close proximity, energy transfer can occur such that the fluorescence is quenched. The quantum yield is reduced significantly so that the fluorescent reporter is non-fluorescent until it's acted upon by some mechanism that is designed into the system. So in this example, using two fluorophores that are connected by a peptide sequence that is specific for an enzyme of choice, the initial agent is non-fluorescent, and then after action by the enzyme, with caspase 3 within a tumor, or other examples are cathepsins or matrix metalloproteinases, the enzyme can be cleaved and then releasing the fluorophores, and fluorescence is restored. This can increase the signal-to-noise ratio because the background is much reduced, and ideally there is not much fluorescence until it is acted upon in the area of interest. And this is an example of an activatable probe that was again made in our lab that also has a radiomedical chelator, and here's a fluorescent probe. This is a fluorophore, an infrared fluorophore, and this is a quenching agent. And because of the close proximity, the agent before activation is almost non-fluorescent. And this image is a fluorescence image with excitation 780 and emission filter of 830 nanometers of just the diagnostic agent, so completely activated. And this agent is the same concentration of dye with the quencher, showing that right after injection there is almost no fluorescence from this agent. And this agent has a DEBD peptide sequence here that is cleavable by caspase 3. Ideally, this would be used for detection of apoptosis once it enters the cell, the tumor cell. But as an initial example of how this multimodal imaging agent can be used, we labeled the agent with copper-64 and filled tubes with the agent, and either the agent with albumin as a control or with caspase 3, and then imaged the mouse, implanted those tubes underneath the skin, and imaged the mouse 2 hours after mixing with the enzyme. Here shows the white light image, and these are all in grayscale to begin with. So the white light image of the mouse, and you can see here pretty easily where the tubes are implanted, and they don't show up on the X-ray. That's not important at this point. But then we took the fluorescence image with this dye in the tubes, and you can see that the tube on the right has much higher fluorescence signals than the tube on the left. Now, if we go to the copper signal, so the radionuclear signal, the images actually look quite the same. So we show that because the scintigraphy is quantitative, the amount of copper on the right tube is the same as the amount of copper on the left tube. So in order to analyze these images, we want to do ROI analysis. So we put regions of interest around the tubes, and we could have selected the region of interest based on the white light, and then copied those over to each one so that we know that we have the same regions in each of the images. And we compare those, the signals, and show that here's the overlaid images of the mice, showing the white light and then the fluorescence signal with a color scale, so that enhances the contrast, showing the fluorescence is much higher in the caspase 3 containing tube than in the albumin control tube. But the radionuclear signal overlaying the X-ray here shows that the control and the caspase 3 have relatively equal amounts of the radionuclide. In fact, the control has a little bit more, it seems, so there may have been a little bit of a difference in the amount of the compound in this tube versus this tube. So by doing the ROI analysis, we can see that the caspase 3 tube had much higher fluorescence, but relatively the same amount of activity. So that we have a higher amount of activity, and we can even use that activity, because we know that that's per molecule, and to look at the tumor, to the activated versus the inactivated ratio, and we've got a ratio of 5.6, so almost 6-fold higher fluorescence in the caspase-containing tube. So, as a summary, no single imaging modality can provide the complete picture of all the biology. So, multimodality imaging can give you a lot better picture of all that is going on in the biology, and can answer a lot more questions in a single imaging session, which will improve the clinical throughput, and in our case, the preclinical throughput, so higher throughput screening and more information. Single molecule multimodality reporters can provide this information as well with a single injection, so that you know the contrast agents are going to the same place and that they're the same concentration, and that can give you more information based on the activation of molecular probes, such as we showed with the caspase 3, or to show that the optical signal is in the same place and the same concentration as the radionuclear signal. A lot of platforms have been developed and are being developed, but imaging platforms that allow easy co-registration of signals, such as the SPECT CT, the PET CT systems that are available. And for optical imaging, the Carestream system and other systems, I believe, that are available. But particularly, as we've shown here, they allow easy co-registration of those signals so that you can have all that signal together, and co-registration of images acquired on different platforms is not necessary. So this can really enhance your throughput. And I'd like to thank the members of the Optical Radiology Lab, particularly Sam Philippou, who is our director, and multiple people have worked on the projects that I described. So I would be happy to answer any questions.
Thank you very much, Dr. Akers. If anyone has any questions, please send them via chat to the box to your right, and we will see if we can get to them. Also, this presentation has been recorded. Probably by the end of the week, we will be sending you links so you can view it at your leisure. If you happen to come in a little late, you can catch the beginning then. Please feel free to use the chat section to your right and send us any questions you may have. Okay, I'm not seeing anything coming in. So if you do have any other questions, I will stay on the line for a little bit longer. You can send them in, and we will respond to you via email. Thank you very much for attending, and have a wonderful week. Again, Dr. Akers, thank you very much.
Thank you.
Goodbye.